Good morning, students. Today we will be studying about the synthesis and degradation of acetylcholine and catecholamines. Let me share the screen. So acetylcholine is a neurotransmitter at the neuromuscular junction at synapses in the ganglia of the visceral motor system and the variety of sites within the central nervous system. Great deal is known about the function of cholinergic transmission at the neuromuscular junction and at the ganglion synapses. The actions are still following in the central nervous system but are not well understood. In the brain, acetylcholine functions the neurotransmitter as well as a neuromodulator. The brain contains a number of cholinergic areas, each of with distinct functions such as playing an important role in arousal, attention, memory, and motivation. Acetylcholine has also been traced in cells of non-neural origins and microbes. Recently, enzymes related to its synthesis, degradation, and cellular uptake have been traced back to early origins of unicellular eukaryotes. So coming to biosynthesis, it is synthesized in the nerve terminals from acetylcholine enzyme A, which is synthesized from glucose and choline in a reaction catalyzed with choline acetyl transferase. The presence of choline acetyl transferase in a neuron is thus a strong indication that acetylcholine is used as one of its transmitters. Choline is present in plasma at a concentration of about 10 millimolar and it is taken up in the cholinergic neurons by a high affinity sodium and choline transporter. About 10,000 molecules of acetylcholine are packaged into each recycle by vesicular acetylcholine transporter and the enzymes. Acetylcholinesterase converts acetylcholine into the inactive metabolites choline and acetyl. That's what is explained here in this figure as well. In contrast to most other small molecule neurotransmitters, the post-synaptic action of acetylcholine at many cholinergic synapses are not terminated by reuptake by a powerful powerful hydrolytic enzyme acetylcholinesterase, which is concentrated in the synaptic cleft, ensuring a rapid decrease in acetylcholine concentration after its release from the presynaptic terminal. Acetylcholinesterase has high catalytic activity and hydrolyzes acetylcholine into acetate and choline. As already mentioned, cholinergic nerve terminals typically contain a high affinity sodium choline transporter that uptakes the choline produced by this acetylcholine hydrolysis. Among the many interesting drugs that interact with this cholinergic enzymes are the organophosphates. They are like usually the pesticides like DDT, then herbicides like 2,4-D, many insecticides, potent chemical warfare agents like serine. So organophosphates can be lethal to humans as well as insects because they have activity the acetylcholinesterase causing acetylcholine to accumulate at these cholinergic synapses. This buildup of acetylcholine depolarizes the postsynaptic cell and renders it refractory to subsequent acetylcholine release, causing, among other effects, neuromuscular paralysis. So acetylcholine metabolism in the cholinergic nerve terminals is being shown in this picture, as I have explained earlier. So now coming to the catecholamines, there are like three main catecholamines. That is the monoamines. It all include three classes of the neurotransmitters. So it's catecholamines, dopamine, norepinephrine, and epinephrine. The indolamines, serotonin and melatonin, and the histamine, which is now considered a neurotransmitter, which is a new neurotransmitter in the past 15 years. The synthesis of these catecholamines, basically your dopamine, norepinephrine, and epinephrine start in the amino acid tyrosine, and tyrosine hydroxylase acts on it, converting it into dopa, and dopa decarboxylates converts it into dopamine, and on dopamine, when dopamine beta hydroxylase acts with it getting converted to norepinephrine, and when phenylethylamine transferase is acting on this norepinephrine, it is getting converted to epinephrine. So, and for the degradation also, catecholamines have a very half-life of few minutes, very small half-life, a few minutes, and they can be degraded either by methylation by COMT, that is catechol, only when transferases or by deamination by monoamine oxidases. So that is what is explained here. Both the enzymes require the cofactors where the COMT uses magnesium as a cofactor, while as a monoamine oxidase uses FAD as a cofactor.